Thank you very much, Kristin. That was great. So I'm going to talk a bit more about the food web modeling component of the guidance. And before I get started on that, I just really high level, I want to talk about when we talk about food web modeling, there's kind of two classes of food web models. There's mechanistic models, that's like your GOBIS style model. It's based typically, they rely really heavily on KOW values to evaluate partitioning. That is not what we're doing here. We're really focusing on empirical values, and we'll start talking about those in more detail. But just so we kind of have a framework of where we're starting from, we're really looking at an empirical model that's focused around lab-based values that measure bioaccumulation from different components of the food web. So, really the question we're trying to answer here is, how can we predict accumulation of PFAS in the food web? Because we need to understand the concentrations in diet items for wildlife in order to estimate their exposure. Oftentimes we can go out and measure this, but for a lot of reasons, cost, logistics, sometimes it's really difficult to go and get enough tissue samples to estimate these exposures. And for most of the other, kind of traditional bioaccumulative compounds that we work with, PCBs, DDT, oftentimes in the past we've really relied heavily on bioaccumulation modeling to get to those estimated concentrations in the food web. And so we really wanted to try and focus our efforts on what we can do now with some modeling parameters to try and start looking at these things. And so as Jason mentioned, we reviewed, a focused data review on, over 125 studies, lots of different bioaccumulation values considered. And ultimately what we came down to was trying to find, specific values that we felt were strong and defensible and developed with meeting certain criteria in the lab, and we could apply in a model in order to move forward and start predicting these things. I'm going to really quickly talk kind of generally about bioaccumulation of PFAS, because unlike some of our more traditional persistent organic pollutants, PFAS don't bioaccumulate by partitioning into lipids like PCBs and DDTs do. And so within the large grouping of PFAS, and even within our small list of 18 out of many compounds that we were focused on, we still see a lot of different partitioning and a lot of different bioaccumulation behavior. So just to make sure we're all kind of working from the same place, the chemical size affects bioaccumulation. When we say chemical size, we really mean that chain length, that in the perfluoroalkyl acids, that perfluorinated chain length. And what we see is that in animal species, so both terrestrial and aquatic animals, we see that the longer the perfluorinated carbon chain, the higher bioaccumulation we see. And this is evidenced, for example, in these fish BCFs. So BCFs are bioconcentration factors. I'll get into a bigger detail on what BCFs are in just a moment. But basically what you see is that they're kind of representing the increase, the amount of bioaccumulation we see for each compound. And as we go up in chain length from 3 to 13, we see a pretty dramatic increase in those BCF values. We see the opposite for plants, though. So the reason that we see that for long chains in animals is because they partition into proteins, and so that long chain is helping it partition better. For plants, what we're actually seeing is that the shorter chain PFAS that are more water soluble, those are being taken up with the water in the plants through the roots, and then as the water evaporates, the PFAS are being left behind. And so what we're seeing actually is you get more accumulation of the shorter chain compounds when it comes to your plants. The other thing that's a really big contributor to how much bioaccumulation potential PFAS has is whether it's a carboxylic acid or a sulfonate. And so what we see is in the carboxylates, those are more bioaccumulative in the plants. They're less bioaccumulative are the sulfonates. So those are your PFAS, PFDS. And it's the opposite again for animals. We see those sulfonates being the higher bioaccumulative ones and the carboxylates being different. So you kind of get these alternating patterns between these two different features. And so it's just kind of something to try and kind of keep in mind as we start thinking about bioaccumulation. So what do you need to do a bioaccumulation model? Well, at the basics, at the really minimum level, you need to understand your concentrations, your abiotic media. And this tool has been developed to really allow that, to allow people to really just focus on, as a starting point at least, concentrations in surface water, sediments, and soils if you have a soil or terrestrial component to your site. We, like Kristin said, we strongly recommend organic carbon data for sediments and soil. we know that PFAS do, especially the long-chain sulfonates, we know that they do partition to organic carbon. It doesn't tell the full partitioning story. There's a lot going on there, but it tells a good enough portion of it that a lot of the bioaccumulation metrics that we're relying on are based on organic carbon normalized values in soil from the lab. And so we really do encourage you to do that. If you don't, there are some, kind of standard assumptions risk assessors like to make. 1 to 2% organic carbon in sediment or soil is about what we see at most places. If it's a wetlands, you might have a higher percentage. So you can make some estimates, and the model will work if you want to, do kind of a estimate for your site. But it's pretty simple analysis, and it's not too costly. So we do encourage people to get it. Tissue data is obviously always really helpful in an ecological risk assessment. If you don't have it, the model is developed to predict it. And what we really, when we've used this model and we kind of, when we think about the best way to apply this model at a site, we really kind of think a phased approach makes a lot of sense here. If you start with your abiotic data and you run it through the model, it'll provide you a lot more information as to which tissue samples maybe are most critical for you sampling. If once you've selected your receptors and you've run this model, you're seeing that maybe your, the bird that eats the benthic invertebrates, your most highest exposed. Well, maybe you want to target benthic invertebrates for your tissue sampling. If it's a piscivorous bird, maybe you want to target your fish. So hopefully, kind of running this in a phased approach can help you make smarter decisions down the road when you start to go out and collect tissue sampling. Because we all know tissue sampling gets kind of expensive, and we want to target, what's going to be smart data to collect and what's going to be most useful. So this is sort of our bioaccumulation modeling framework, and I know there's a lot on this slide. I'm going to break it down step by step in just a second for everyone, but there's a couple terms on here I want to make sure everyone's aware of. BCFs, BMFs, and BSAFs. So bioconcentration factors. Bioconcentration factors are the ratio of your concentration in your organism to that of its respiratory media. Generally, we mean water. But it can be air. You can have a BCF based on air. It's pretty rare, though. Usually we mean water, a fish or an invertebrate respiring that water. We're not really talking about BAFs too much here, but we do discuss it in the guidance. The difference between a BCF and a BAF, often confused, is that a BAF is a field-based BCF. And the reason we distinguish between the two is that it's still based on the ratio between your organism and your respiratory media. So you've gone out and you've collected a water sample and a fish sample in the field, and you're using those to calculate a BAF. But that fish really isn't just being exposed to water. It's going to be exposed partly through its diet as well, and you're usually not accounting for that when you measure a BAF in the field. And so we try and distinguish the two. A BCF has come from a lab where the only exposure media was that respiratory media. BAFs are field-based, a little bit more uncertainty around those values. BMFs is the ratio of concentration of an organism to that of its diet. Oftentimes this is done through spiked diet dosing studies in a lab so that you can understand how much of that, the component of that PFAS in its diet that's actually being taken up into the organism. And lastly, BSAFs, that S can be soil or sediment. And generally what we mean there is the concentration in soil or sediment, the ratio of the concentration in the organism to that of soil or sediment. This is usually used for benthic invertebrates or terrestrial invertebrates like worms. So now that we're all kind of talking the same language, what the model does is it really takes what we felt were the best available BCFs, BSAFs, and BMFs and applies them in a stepwise way to get to a proposed estimated diet item for those organisms. So first step, taking a look at benthic invertebrates. You've got your concentration of PFAS in sediment. Because the BSAFs that we decided were the best ones we wanted to use were based on organic carbon normalized values in sediments. During the laboratory procedures, we recommend you use your organic carbon normalized sediment concentrations for this. And then you multiply the BSAF is essentially multiplying that organic carbon normalized concentration in sediment to predict the concentration in benthic invertebrates. You also have pelagic invertebrates, primarily exposed through water. So what we do in the model is we take your concentration of PFAS in water, multiply it by some BCFs that we've selected or recommended for pelagic invertebrates, and those are then used to predict the concentration in the pelagic invertebrate. Same idea for aquatic plants, really. We've got BCFs for aquatic plants that we've recommended through the literature review, and you're taking the concentration of PFAS in water, multiplying it by that BCF to get a predicted value in your plant. All really kind of simple one-step modeling components here. kind of, just getting to the basic components of the fish food web at this point, really. We're just starting to get to, really kind of low-level trophic, benthic invertebrate, pelagic invertebrate, and fish. Starts to get a little more complicated when we start looking at fish. So fish really are the sum of their exposures through water and diet. So the model is taking the sum of your estimated concentration in water times the BSAF drive for fish, the concentration in plants times how much of their diet is plant. So if they're eating 50% plant, you're multiplying it by a factor of 0.5. If it's 30%, it's 0.3, times that BMF for fish. And BMFs generally are not distinguished between the diet item. The BMF for fish was based on a feed that was provided in the lab study. And so we apply the same BMF for whatever that diet item is for fish. And then so it's the same thing, your concentration in your pelagic invertebrate, which was predicted in the previous step using the BCF and the concentration in water. And then you're multiplying that by the BMF and the proportion of the pelagic invertebrate's diet for that fish. Same thing for benthic invertebrates. You've predicted the concentration in benthic invertebrates in the first step of the model. In this next step, it's being used with the proportion of the benthic invertebrate in the diet and the BMF for the fish. So in the user tool, these components are all sort of up to the user to specify. We have some kind of recommended default values in there, but this is really kind of where that ecological risk assessor you're working with or you are needs to kind of look at your fish species and say, well, this is what we think is most likely their, for the most representative fish species at our site, this is what their diet tends to look like. And then for predatory fish, it really works through the same, those first few components are the same. Your BCF for the water uptake, plant uptake, and pelagic and benthic invertebrate uptake. But then we've also worked in a component for that forage fish uptake because, of course, big fish eats little fish, right? And so that sort of takes you through the aquatic component of the bioaccumulation model. The terrestrial side of it is a little bit more simplified at this point. It's really just taking your organic carbon concentrations in soil and multiplying them by the BSAFs for terrestrial invertebrates or the BAFs for plants to get to those concentrations in diet items on the terrestrial component side. And as Kristin mentioned, we're working on an Excel tool for the terrestrial side of this as well, so stay tuned for that. So we have a lot of BMFs and BAFs and BCFs and all those fun things. We did a pretty deep dive data review on this, and we did have a few specific criteria we wanted to look at. Primarily, the documentation of steady state. So this is not a time-dependent model. This is a steady state assumed model. It was really important to us that the concentration in that exposure media was measured, not just nominal, but also that it was, stable throughout the course of the exposure so that when you're calculating that BAF from that ratio, you have a good understanding of what that exposure media was. We really looked at using spike studies because of the homogeneity in dosing of PFAS, known composition of PFAS, so knowing that the linear PFAS was used instead of branched, which tends to be more conservative because it tends to be more bioaccumulative, whether or not there were precursors present, having a good understanding of all of those. And also, freshly spiked studies, they do tend to be more bioavailable compared to field studies or, PFAS that's been in AFFF that's been sitting in soil for the last 60 years. You get more bioaccumulation. And so, we did want this model to have a bit of a conservative lean to it. And so we generally selected for lab studies that had that spiked media. And where we saw, 2 lab studies with both similar design and both really good quality, we did err on the side of conservatism and select the higher bioaccumulation value. So ultimately, what you get in the guidance is tables and figures that recommend a whole bunch of BCF and BAF and BMF values. So we've got water to pelagic invertebrate bioconcentration, sediment to benthic invertebrate biota sediment accumulation factors, water to aquatic plant and water to fish tissue bioconcentration factors, diet to fish tissue biomagnification factors, and then the 2 soil to terrestrial invertebrates and terrestrial plants. So this is that same example, now that I've explained what a BCF is. This might make a little more sense. But this is what we provided in the guidance for fish BCF values. And so what you can see here, again, we see that increasing accumulation with those longer chain lengths. If you look and compare sort of a similar chain length, carboxylate versus sulfonate, you see that those sulfonate values are generally on the higher side. And so you'll find, for all of the bioaccumulation values or types of parameters we've discussed, you'll see similar graphs and tables and figures like this in the guidance document to help outline, what our recommended values are. Similarly, for benthic invertebrates, this is another example. Here again, we see, that increasing accumulation through many of the PFCAs with increasing long chain length. We see some tapering out there at the longer chain, which we thought was interesting. Same with the sulfonates as well. But of course, we don't have values for everything. Can't ever. Not every study measures all 18 of our PFAS, unfortunately. And so we did have some data gaps. And we really wanted to provide some options. We understand that there's a lot of best professional judgment that goes into the use of surrogates or the use of filling data gaps with other information. And so, we wanted to make some recommendations on what we thought was the best approach. Certainly, it's not the only approach, and this is really just our recommendations. But what we went ahead and did in the model tool is an option to apply surrogate values where we had data gaps. And we generally base these on QSARs, which are quantitative structure activity relationships. And really what we mean here is the where we had data where we could correlate along those chain lengths for the carboxylates or the sulfonates separately, we used those and tried to apply those values. And so a lot of best professional judgment went into selecting these. For example, when we were looking at our aquatic plant BCFs, we had data, all of these hollow circles are the actual lab data. And then we were missing a value for PFBA. We kind of looked at that graph and said, well, if you see the way they're kind of tapering off, I think we can probably most likely apply the, next carboxylate up, the PEA value, and provide that for the 3-carbon chain PFCA. And so that's how we extrapolated there. That's an example of that. For the example for the pelagic invertebrates, the BCFPIs, there we had data for 5 of the PFCAs, and we took that relationship. It had a strong relationship with carbon chain length, and so we extrapolated that relationship out both above and below to represent the data gaps we had. Like I said, the use of surrogates, it adds a lot of uncertainty. We felt it was a good option to be able to fill in those data gaps, but it really should be done carefully and considered carefully before using as a basis for any significant risk management decisions. If you're using surrogate values to make big decisions about going out and dredging or cleaning up a site, you really need to think very carefully about what the uncertainty is around there. So just keep that in mind. So we have, done a little bit of testing of this model. We do like to think it works fairly well. We did some of the preliminary model frameworks we had working with Emily Larson in 2018. We put together sort of a comparison of 5 example AFFF sites using a preliminary version of this model. And we found that it did have good agreement with fish tissue data where we had measured data to compare it to, usually within about a factor of 5. And so that's pretty good agreement for a model like this. We did some sensitivity analysis, and we did see that our parameters tended to err on the more conservative side than some of the field measured values. Again, that relates to kind of that level of control in a lab study, along with typically that higher availability of freshly spiked PFAS in those media. But we are also looking for more opportunities to test and validate this model. So if you've got a data set that you're working with and you want to test out and see how your measured concentrations in fish compare to those that the model predicts, we'd love to hear from you. So please don't be afraid to reach out with thoughts on that. And lastly, we really did want to make this guidance document generally something that can be broadly applied. This is not just like Kristin, Jason, and Jen's rules for eco risk. This is very much, pick what works for your site. And so for each of the kind of parameters we've been looking at, we've included an appendix that provides all of the studies that we looked at. And in many cases, there may be a species that was a field-based study that you think is more representative of the species at your site, or another parameter that you thought was more important for your site specifically. So we encourage you, if you don't agree with what we've recommended in the guidance document, check out the appendices, see if there's something that you think is better there. They really are meant to be useful for everyone. So once you've gone ahead and predicted all of these concentrations in your food web, and you've got estimated concentrations in plants and invertebrates and fish, Well, then we go ahead and estimate our concentration, we estimate our total daily intake for wildlife. And this is where those exposure factors that Kristin was talking about in your selection of wildlife receptors comes into play. It's a standard approach that's pretty much used for any chemical. This is right out of, kind of eco-rags. And really, it's estimating your daily dietary intake for wildlife using the predicted or measured concentrations if you have it. And really, your intake for your wildlife is a function of the concentrations in what they're eating, how much of those diet items they're eating, what their food ingestion rates are, incidental ingestion of soils or sediment along with surface water, and then their body weight and their site fidelity, that area use factor, which is always a critical thing to think about as an ecological risk assessor, and always often up for debate. But really, this is all pretty standard exposure modeling for ecological risk assessments at this point.